Dear colleagues, today I would like to talk about the nervous tissue. In my talk, I would like to run through very fast on the review of the basic tissues. I would like to list them, that you can place this basic tissue into the system of basic tissues. Then I would like to highlight some really basic terms regarding the nervous system. Many people are, I hope, already familiar with these terms, but just to make sure that everyone knows these things, I would like to revise them. Then we will turn to the nervous tissue proper. I will discuss some basics on the histological features of neurons. I will tell you the classifications also. And then after this introduction, we will turn to the sensory ganglion and the autonomic ganglion, which are slides, what you get familiar with in the practices. And very briefly, I would like to summarize those things, what you have to find in the sections, the preparations also as well. So let's turn to the revision of the basic tissues very fast. Just to list them, in the first couple of weeks in this semester you studied the epithelial tissues, several kinds of epithelial tissues you studied with many functions. Then you turn to the family of the connective and supportive tissues. This was the second basic tissue group, and then the muscle tissues came with cardiac muscle, smooth muscle, and skeletal muscle proper. And then the fourth basic tissue, after these three, what you already should know, is the nervous tissue, and this is what I have to talk about. This is the first lecture about this topic, and then I will also give another one to cover this quite complex field. Then the next part of the talk is about basic terms, as I mentioned. So you must know these, but just to make sure we revise them for a moment. So this scheme shows you the parts of the nervous system, and if you want to start somewhere, you have to define the central nervous system. The central nervous system is comprised by the brain and the spinal cord. Here you are. And then outside the brain and the spinal cord, the nervous tissue continues also into the periphery, that you can call peripheral nervous system. In the peripheral nervous system you find the nerves. Also you may call them peripheral nerves, but they are already in the periphery. And then we may classify them into cranial nerves and spinal nerves. In the anatomy studies in the first semester, you are most of the times familiar with the spinal nerves, but when you study the skull, you will at least study the names of the cranial nerves also, which directly arise from the brain. Spinal nerves arise from the spinal cord, of course, as the name already suggests. And in the periphery, besides the nerves, we also find ganglia. They are related to the peripheral nerves, and we may classify them as sensory and autonomic ganglia. At the end part of this talk, I will come back to the details in these types of tissues. Next, we stay at the basic terms, and after the anatomical classification of the nervous system that we have central and peripheral, I would like to turn to the functional classification. And based on the function, we can split the nervous system into somatic and autonomic nervous system. Let's start with the somatic one. In the somatic nervous system, the signal comes from the environment, and the somatic nervous system picks up this signal from the periphery. It will collect the stimuli from the environment. It will translate it into electric signals, and finally it is then transported to the highest order centers, and this is the sensory part of the somatic nervous system. And after this, we make a decision, and then these decisions will be performed, should be performed, and therefore the signal has to go now in opposite direction. It has to reach the periphery, and this part is the efferent or motor part, which finally performs the function. It carries out the movement. Just an example that you understand. You want to have a coffee. You make a coffee. You grab the cup, which is pretty hot, and you have to make a decision. Either you put it down or you hold it. If it's too hot, you have to make a decision. because if you hold them up for a longer period of time, you get some serious injury on your skin. The sensory part will inform you about the high temperature, and then if you decide to drop the mug to stretch your fingers, the command is performed through the other part, the motor part of the nervous system. How does this work? It's a very simple scheme here. This is the body, the white area, the green represents the central nervous system with the brain and the spinal cord, and here you have the peripheral nervous system, that should be the yellow box. Now the signal, the stimuli comes from the environment. It might be high temperature or mechanical, chemical, visual, acoustic, and several other kinds of stimuli may come. And this stimulus comes to the receptor, which translates the stimulus into electric signals, and then either through spinal nerves or through cranial nerves, the information arrives to the brain or to the spinal cord to perform something, to carry out something, and then now the higher order centers know what happened, and this is the afferent direction or sensory information processing. Then the brain makes a decision, and from the spinal cord or straight from the brain to the periphery, through the spinal nerves or the cranial nerves, the signal comes to the effectors which carry out those commands which the brain elaborates, and then you perform the movement. Most of the times muscles are responsible, of course, for the answer to the environmental challenge, but in some cases you will see that even glands may be activated. But this already belongs to the autonomic nervous system and not to the somatic one, because this part, the somatic nervous system, is the conscious part of the nervous system, where we get informed about the environment at the level of consciousness. In contrast, the autonomic nervous system is independent from our decisions. That's why it's called autonomic nervous system, because we do not have a voluntary control on this system. It is also divided into an afferent part. This perceives against stimuli, but typically from internal organs, from inside. For instance, oxygen content of the blood, or pH levels, or the movement of the internal organs, for instance, are stimuli which come to the brain, but they never reach the level of consciousness, so you don't know your blood pressure currently, or you don't feel the oxygen saturation of your blood. And then the brain measures these internal organ functions. It is responsible for the maintenance of the body's main functions, and if anything has to change, then the efferent part can perform what the brain has to control, and then you have here involuntary functions to control, typically, for example, smooth muscle function. So if the blood pressure is low, one can perform a muscle contraction in the walls of the blood vessels, the smooth muscle contracts and the blood pressure increases, or you can control the saliva production in the glands, or increase the heart rate by changing the contractions of the cardiac muscles by accelerating them, for instance. This is controlled by the different parts of the autonomic nervous system, by the sympathetic, parasympathetic, and also the enteric nervous system. The sympathetic one gets activated if you are in a stressful environment, in an exam situation, for example. But if you pass the exam and you get relaxed, then the parasympathetic nervous system is on, and that will load up the energy storages, the heart rate decreases. So these systems are antagonistic. The enteric nervous system controls the function of the internal organs. I don't have time to go into those details, but in later studies you will hear about this also a lot. After this, I would like to turn to the nervous tissue. So in the next slides, I would like to show you some basics on the nervous tissue proper. First of all, the nervous tissue you can divide into two main groups, two main races of cells. First of all, we have the neurons or nerve cells, and we have also the glial cells. I will come back to this point. But first about the nervous tissue, about the neurons, the nerve cells. Nerve cells are responsible and specialized for the perception, transmission, processing, and relaying the electric signals. This is an important sentence, an important definition, and probably the most important part is this here, specialized for. So basically, many kinds of basic tissues, the cells of those are able to perceive signals, to process them, to relay them, but they are not specialized for this function. This is the key feature of the neurons, that they are specialized for these functions. Therefore, the greatest variety of biological functions are controlled by the nerve cells. This is why the nervous tissue is the most important regulator of the human body. Nerve cells communicate with each other. This communication goes through synapses most of the times, and not only the nerve cells with each other, but even with other kinds of tissues, with muscles, for instance, or with glands. The nervous tissue can communicate. About synapses, you will hear lectures in the neuroanatomy series of lectures. As I mentioned, the nervous tissue is composed by two main races of cells, neurons and glial cells. We don't talk too much about the glial cells today, but we have to mention that they are found, of course, both in the central nervous system and also in the periphery. But the central glial cells we do not discuss in this subject, but we will restrict our description to the peripheral glial cells. Today I will talk a bit about the satellite cells, and Schwann cells will be discussed in my next talk. One can classify the neurons, and if we would like to classify the neurons, one can turn to the functional classification. So we can classify the neurons according to their functions. This comes back to the point what I started with the sensory part of the nervous system, because we have neurons which pick up the signal from the periphery. They are the afferent neurons, or sensory neurons. They transmit the information to the towards the higher order centers of the central nervous system. In the central nervous system we have interneurons. They connect different cells with each other, and they allow the communication inside the central nervous system. And finally, we have motor neurons. They are responsible for performing the functions to control the movements, for example, but they also may control some autonomic functions, as you will see soon. And if I would like to talk about neurons, and this is a histology talk, I have to show you the basic histological features of the neurons. This is the cell body of a neuron. One can call it soma or perikaryon. In the center, the nucleus of the cell is situated, which typically has a well-developed pronounced nucleolus. And the cell body here, also called soma or perikaryon, has a couple of processes which we can call dendrites. These dendrites divide multiple times. They are like branches of a tree. That is the origin of the nomenclature of the name also. Each neuron has one single axon, which is typically longer than the dendrites. The axon rises from a specific area, which is called axon hillock. This is the place where the action potentials are formed, and this is an important spot for the electrophysiological functions of the cell. The cell membrane contains specific ion channels here. The axon, on its way, splits sometimes into branches. They are called axon collaterals. The division is typically perpendicular. And finally, the axon ends in a so-called telodendrion or end tree, and this telodendrion allows the communication with the next cells. So dendrites and the cell body forms synapses. It picks up the signal from other cells, and this neuron then will perceive the signal, it will change the signal, and then after this it may even give the signal further to the next cell, which will form another synapse through which then the signal goes to the next neuron, for example. After this, what I would like to tell you is again a classification of neurons, but not the functional, but the so-called morphological classification of neurons. In my last slide, I showed you a neuron with many processes, and that's typically the most common type of neuron, but not always you can find in your slides these neurons. You may see sometimes so-called unipolar neurons. They just have one single axon. They are very primitive nerve cells. We see them very rarely, and they do not belong to this semester stuff. Then we go to the bipolar neuron. They are a little bit more complex in structure. They have a dendrite and an axon already. They are also not very common. One can find these cells in the sensory organs typically. And then what I would like to detail a little bit more is the so-called pseudo-unipolar type of neuron. The name refers to the fact that it has one single process that after a very short distance splits into two branches, so it's a fake unipolar, pseudo-unipolar neuron, which has principally one process towards the periphery, one that goes to the center. So one may call them peripheral central processes, but if we zoom in with an electron microscope, it turns out that both are more axons in structure, and because of their different function, one can be called dendritic axon, which picks up the signal, and the other one is the axonic axon, which gives the signal further to the next cell. The example for that is the sensory ganglion, and I will come back to this point at the very end of this lecture. And finally, we have the multipolar neuron, that typical neuron what I showed you in my last slide. You have more than one, but most of the times many dendrites. There is one axon which goes to a far point most of the times, and most of the cells in the central nervous system are of multipolar type. And today I will finish my lecture with the structure of the autonomic ganglion, where you can observe again multipolar neurons in the peripheral nervous system. This scheme shows you a part of the central nervous system, the cross-section of the spinal cord. I don't want to talk about the central nervous system, but some basic terms I really need to make you familiar with this story. In the spinal cord, this butterfly-shaped area is the gray substance or gray matter, and then outside we have the white substance or white matter. That is the place where the nerve fibers travel. They comprise the ascending and the descending pathways of the central nervous system. I don't want to go into those details now. I just have to mention the gray matter, because this is the place where the neurons, the cell bodies, the pericardia of the neurons are found. So in this scheme I would like to explain how the information goes and how is this related to the cells I discussed with you in the earlier slide. So this is the skin, and the stimulus comes from the environment. The pain is there. You cut your finger with a knife, or you pick up a hot object, a coffee, which is in a very hot cup, and that burns your skin, and the receptor in your skin for the pain gets activated. And this neuron with its peripheral process picks up the signal and carries it to the central nervous system. The neuron has a pseudo-unipolar shape, and the neuron is with its cell body located in the periphery. And if we are in the periphery and we have multiple cell bodies, neurons in the periphery, and they have a sensory function because they pick up the signal, we are talking about a sensory ganglion. So it is made up by pseudo-unipolar neurons, and the function is the afferent or sensory information processing. Once again, I would like to highlight that this is a ganglion. What is a ganglion? This is an important definition to be studied. A group of pericardia of neurons outside the central nervous system, shortly in the periphery. So not in the spinal cord, but outside the spinal cord, a group of neurons is found. Then the signal is conveyed to an interneuron. That signalizes further to a motor neuron. And now, in the periphery, you can perform something, you can do something to avoid the pain. You can release the hot object, the coffee, and in order to perform that, you have to stretch your fingers, you have to contract your muscles, and the signal now from the motor neuron comes back to the periphery, and in this case, the information goes in an efferent way. It's a motor way that lets the muscle contract, and then you can drop off the mug that it doesn't burn your skin. So this is how the information spreads, and I wanted to explain in this slide the sensory ganglion's significance. And besides this, we have the autonomic nervous system, because that was a somatic. Now we turn to the autonomic nervous system, and in the lateral horn of the spinal cord, there is a multipolar neuron here, which goes with its preganglionic process to the periphery and synapses to the multipolar nerve cell. And this multipolar nerve cell with its postganglionic fiber will innervate the organ. For instance, if in a stressful situation you have to increase your heart rate, this neuron in the spinal cord fires. Through the preganglionic fiber, the signal goes to the periphery, and through this synapse, it will activate a neuron that goes already to the heart, and the synapse is the place where the signal continues from the preganglionic fiber to the postganglionic neuron. And then this neuron is again in the periphery, and it's not alone. You have a couple of cells in your group that one can call autonomic ganglion. And the autonomic ganglion is again a ganglion, so please memorize the definition for the ganglion. It's a group of pericardia outside the central nervous system, and that fits with the definition also. Before I go to the neurons, one more remark, but I will come back to this point in my next talk. The nerve fibers traveling from the periphery to the higher order centers, or from the center to the periphery, are bundled, and these are the peripheral nerves. I will come back to this point next time. And then this slide is a very nice picture that summarizes some basic terms related to the neuron. So if you look at this picture and you review these terms on the picture, you have a quite strong overview, visual overview on the basic terms related to the neurons. What I would like to highlight now is the light microscopic picture of the neuron. This is a missile-stained preparation from the motoneurons of the spinal cord, and this one is also new motoneuron in the spinal cord with hematoxylin staining. Please note that the nucleus is euchromatic. You have a well-developed nucleolus here, which is visible in hematoxylin also, and the cytoplasm is full with rough endoplasmic reticulum, which is called in missile-stained missile bodies. And then here you can see the rough endoplasmic reticulum, which is responsible for the basophilic appearance of the neurons in the hematoxylin stained preparation. And in the last part of my lecture, I would like to introduce you the sensory ganglion. The sensory ganglion, if I want to describe it histologically, it is composed by sensory neurons, pseudo-unipolar neurons, and in a hematoxylin stained preparation, you can see large pericardia. They are around 50 to 100 micrometer in diameter even. You can see a euchromatic nucleus with a well-recognizable nucleolus in the center of the cell. And in hematoxylin stained preparation, please note that the cells have a strong basophilic appearance because of the before mentioned rough endoplasmic reticulum. And also it has to be mentioned that the cells are quite long-living cells. They do not regenerate, so they have a long lifespan. Therefore, typically they store a substance called lipofuscin, or aging pigment, which is related to the renewal of the membrane structures in the cell, and some inclusions occur in the cell which have a brownish color, and that is recognizable also in many of our preparations. Besides the neurons, as I mentioned earlier, the nervous tissue is composed not only by neurons but also by glial cells. And around the cell bodies of the neurons, one can observe the so-called satellite cells. They belong, as I mentioned earlier, to the group of the peripheral glial cells. And please note that they have a supportive function. Not only mechanically supportive, but also nutritive functions they have. So they kind of serve the functions of the neurons in many aspects. They protect them, they feed them, they surround them. Besides these, next to the neurons, we also can observe nerve fibers. The structure of the nerve fiber will be discussed next time. I just would like to mention now that the nerve fibers are covered by Schwann cells, and these are the nuclei of the Schwann cells, but regarding the functions and histological features, I will give you another talk later. If I stain the same tissue sample with silver ions, seemingly different picture appears, but if you study these cells, it turns out that they are quite similar to those. Some shrinkage occurs always, but if you look at the cell, only one single process arises from the cell that makes several loops around the cell body. It is wrapping around the cell body, and then somewhere it splits into two branches. So that proves that indeed you have here in your slide a group of pseudo-unipolar neurons, which proves the definition, the diagnosis that you are in a sensory ganglion in this preparation. So the dark black slide, if you zoom in with a high power magnification, you can see these loops around the cell body and one single process in the cell. You can be sure it's a sensory ganglion. And that has to be compared with the structure of the autonomic ganglion. This will be the next. So until now I was talking about the sensory ganglion, but now we turn to the autonomic ganglion, which is again a group of neurons outside the central nervous system, but the cells have a different shape. And indeed, if you use a silver nitrate strained sample, and if you check one single cell here, for instance, if you count, you can see that at least 10 processes, 10 dendritic processes arise from this single cell body, which proves that you are in a ganglion which is formed by multipolar neurons. And if you have multipolar neurons, that fits with the definition autonomic ganglion. Please do not forget that these multipolar neurons form synapses, since we have dendrites everywhere, but very tiny axons can be found also in the preparation, and with electron microscope one could find synapses between those. And in these synapses, the most important neurotransmitter, which carries the signal from the preganglionic fiber to the cell, is the acetylcholine, which is an important neurotransmitter and a common neurotransmitter in the peripheral nervous system. And also in the autonomic ganglion you have cholinergic synapses. So the time is over. Thank you for your attention. And next time I will continue with the structure of the peripheral nerve and that of the nerve fibers. Thank you for your attention.